No. Hello, everybody.
Why does it say September 23rd for the exam?
What does it say before the exam? It's a review
Oh,
session.
it's a review. Okay, okay.
Okay.
My bad. Sorry.
Maybe I should change this. Wait. Um, guess it's confusing. Need to take this out. Okay, I guess that's better. Okay. Okay, so on Tuesday, September 23rd, we'll have a review session. It's not going to be recorded because what would be the purpose of have a review session if no one shows up because they think it's going to be recorded? So no, you have to be there. It's class time, so it's there's no class on that day, so you should all be free because that's the scheduled class when we take the class. Okay, so so Tuesday, September 23rd, we have a review session from 4 to 5:15, and I hope that there will be lots of students attending. If you cannot attend, you can always ask a colleague. It's online, it's on Zoom, yes. I know that some of you have a Jewish holiday, but I don't know how. If you have any suggestions, just email me of how we can work out a solution for that. Okay, so I also posted on our Brightspace website. I posted material and practice questions for lecture exam 1. So there's question examples. You just have to click this when you go to the announcement. So when you open up the Brightspace, you will see this announcement right away. And you have examples for questions and also the material that's going to be covered in the exam. Okay, so those of you that cannot attend the review session because of religious reasons, please send me an email and maybe we can work something out. Okay? Okay, so we have lots to cover today. In the last class, we talked about protein synthesis, transcription that occurs in the nucleus and results in the production or synthesis of messenger RNA, the mature messenger RNA. Then that messenger RNA moves to the cytoplasm, binds to the ribosome, and then this interaction between the messenger RNA and the transfer RNA allows for the protein to be synthesized one amino acid at a time. So two amino acids will bind to each other through a peptide bond that's formed in the large subunit of the ribosome. Now, the review will not be recorded and will not be posted because it's during class time, so everybody should be available. And if it would be recorded, who would show up, right? There would be one person maybe showing up, or no one would show up because you would think it's going to be recorded. So this is a class participation. Okay, so you can go here and look at the questions and also at the material that's going to be covered. So today we're going to study or discuss DNA replication. So this is another mechanism that happens in the nucleus. So in the nucleus, you have the synthesis of messenger RNA and DNA replication. So when does DNA replication occur? So DNA replication means that from one molecule of DNA, you make two identical DNA molecules. And this is necessary because of cell division. So from one cell, you have two cells by mitosis or four cells by meiosis, and they need to contain the full amount of genes so that are and the full amount of DNA. So you need to make two copies of DNA during DNA replication. So when does this occur? So here is represented, this circle represents the cell cycle of a cell. So it has G1, S, G2, and mitosis phase. So we don't have here represented meiosis. So G1 and G2 are growth phases. So this is when the cell gets a lot of nutrients and prepares to the other two phases, which are highly demanding. So the S phase is where DNA replication occurs. So S is easy to remember because it represents synthesis of two molecules of DNA from one template. You have G1, which is a growth phase, and then the S phase where DNA is replicated. So now you have two molecules of DNA, then the G2 phase, and then mitosis, which allows for one cell to go through mitosis and give rise to two cells. Okay, so if you have questions, now I would like to focus on the material. If you have questions regarding if there are classes or no classes, there's no classes on Monday, Tuesday, and Wednesday. So we can address this at the end of the class. Okay, so what does it mean, DNA replication? So there's ways that DNA could be replicated. Either a conservative way, a conservative manner, where you would have the initial DNA molecule, and after it being replicated, this would give rise of a, let's say, an old DNA molecule, and then the newly synthesized DNA molecule. So the one DNA molecule would contain the strands from the original DNA, and the new molecule would contain the two newly synthesized strands. But this is not what happens. What happens is what we call a semi-conservative manner in which the DNA, by which the DNA replicates. So what happens is these are DNA is a double strand molecule. So what has to happen, the two strands have to be separated. Remember that they are linked by hydrogen bonds that link the bases, the purine and pyrimidine bases. So there are enzymes that break apart the hydrogen bonds, and then you have the two DNA strands separated, and then by a process known as DNA replication, we have a copy of each of the strands, and the new copy binds in a it's a semi-conservative process, because each new DNA molecule contains one strand from the original molecule and one strand that is newly synthesized. So you see here that it's a mixture of new strand and the original strand. So this is what we say a semi-conservative molecule is formed. So it's a hybrid. That's a very good way of describing it. It's a hybrid. Why? Because it has the original strand, here represented in gray, and it has the new strand that is newly synthesized. And each of the two strands, each of them is made up of the original strand and the newly synthesized strand. Okay, so how does this occur? So this is a very, very complex process. Remember that the molecule that's 6 foot long has to be copied into two exact copies of the same molecule. So, and this is all done not by a photocopy machine where you put a sheet of paper and you can get two identical sheets of paper. This is a very complex process, and the DNA molecule is replicated not all at once, in sections. Okay, so the first thing that forms at the DNA molecule is the replication forks. What does that mean? That means that the strands, the two DNA strands, are separated. Remember, they are linked by hydrogen bonds. So these replication forks, there's many of these that are formed along the DNA molecule. So this is the parental molecule. And now remember that these numbers, 5' and 3', 3' and 5', these are very important numbers, because this tells you the direction of the replication. And remember this, in a little while we'll come back to this. So the first thing that happens is the formation of this fork. And the enzyme, there are many enzymes involved in this process of DNA replication, but the major enzyme is the DNA polymerase. So this enzyme, just like the RNA polymerase, was able to connect two nucleotides through a phosphodiesterase bond to make messenger RNA. That was the RNA polymerase. Now you have the DNA polymerase that is able to form, plays a really important role in DNA replication, because it also makes the phosphodiesterase bond between two nucleotides. Okay, So here are some of the enzymes involved in this process. Helicase. This enzyme breaks apart the hydrogen bonds. So it allows the separation. Then you have single strand DNA binding proteins. What is the function of this? These proteins will bind. So here you have the DNA helicase that breaks apart the two strands. So this here is the double strand DNA. So after the helicase functions, you see that the two strands are separated. And here are these binding proteins, single strand DNA binding proteins, that very helpful maintain the two strands apart. Then there's another enzyme, the topoisomerase. What is its function? Its function, it prevents. So if you open up something that is attached to each other in a helical fashion, you're going to induce the high supercoiling, so more and more binding of the two double strands. So you don't want that to occur. That's called the supercoiling. So if the DNA would be more and more attached and forming this highly dense molecule, they would be very hard to replicate. So the topoisomerase prevents that supercoiling of the DNA molecule. So here we have three proteins, two enzymes, the helicase and the topoisomerase, and the single-strand DNA proteins to keep the DNA strands separated. Okay, so then what happens next? So the DNA polymerase has a very special requirement. It cannot start functioning if there are not some RNA primers placed on the DNA at the initiation site. So we have to have RNA primers. So these are small segments of RNA that are synthesized at the initiation site where the DNA polymerase is going to start replicate DNA. So this is a requirement. So these RNA primers are made by the DNA primase. So this DNA primase, its function is to synthesize these RNA primers that are located at the initial site of DNA replication. So this is a requirement. The RNA polymerase did not have this requirement, but the DNA polymerase does. So you need to start this process of DNA replication. You need some small portions or fragments of RNA to be placed at the start site. So this is carried out by the RNA primase. Primase, if you remember, is the enzyme that will synthesize the RNA primers. Okay, so now we have everything prepared. We have the two double strands separated by the helicase. You have the single strand binding protein separating the strands, and you have the topoisomerase that is preventing supercoiling. Okay, and now we have the DNA primase that has synthesized and attached the RNA primers. So now the scenario is it's ready for replication. So now this is done by the RNA polymerase. So now the DNA polymerase, not the RNA, DNA polymerase. So DNA polymerase will start synthesizing the new DNA molecule in this 5' 3' direction. And I will explain why this is important. So The steps are first, open up the strand and add these proteins to keep the strand separated, prevent the supercoiling with the topoisomerase, and make these RNA primers. Now everything is settled for the DNA polymerase to start synthesizing a new DNA strand. Okay, now it's very important. Wait 1 second. I skipped one of the slides that I want to show. Just 1 minute. This is the one. So let me go back and go here. Okay, so we're back. Let me do this. And let me do this. Okay, so we were talking about how the DNA primase sets up the RNA primers that allow the DNA polymerase to start replicating one strand. Now, it's very important to remember that this DNA polymerase, which is absolutely dependent on these RNA primers. Now, it's going to synthesize the new strand in the 5', 3' direction. What does that mean? That means, so here's a nucleotide. Remember that the nucleotide is the monomer of RNA or DNA. So these nucleotides are going to be attached one to each other through a phosphodiester bond. An amino acid gets attached to each other by a peptide bond. Here you have a nucleotide attached to each other by a phosphodiester bond. Okay, now let's look at the nucleotide. It has a sugar. In the case of DNA, it's deoxyribose. Then you have a base that can be A, D, C, G. And then you have a phosphate group. Now, notice here that the carbons are labeled by numbers. So this carbon into which the base attaches is 1'. Then you have 2', then you have 3', 4', and here's the 5'. So there's 4 carbons here involved in this nucleotide. 1', 2', 3', 4', 5'. So what does it mean that the DNA polymerase forms or binds or replicates the new strand in the 5', 3' direction? Why? Because the phosphodiester bond, so here's one nucleotide, here's another nucleotide. And where does this phosphodiester bond occur? So the phosphodiester bond is between the carbon in the 5' position of one nucleotide and the carbon in the 3' position of the next nucleotide. So that's how the DNA polymerase links the two nucleotides. So it uses the carbon in the 5' position and links it through a phosphodiester bond to the 3' carbon in the next nucleotide. So that's why the DNA polymerase always replicates the DNA, the new strand, from the 5' to the 3'.
Professor, do you mind repeating that? Why it starts from the 5'?
So remember, this carbon here is the 5' carbon, right? 1, 2, 3, 4, 5. And this 5' carbon of this nucleotide is going to be attached to the 3' carbon, the 3' carbon of the next nucleotide. That's how the DNA polymerase, due to its folding and so on, that's how it can generate this phosphodiester bond that links the two nucleotides. Okay, so the DNA polymerase will synthesize the new strand in the 5' to the 3' direction. So 5' to the 3' direction. These are the two nucleotides. Here, the phosphodiester bond that is formed between the 5' carbon of one nucleotide and the 3' carbon of the next nucleotide. So that means that the DNA replication is directional. The DNA polymerase synthesizes a new strand in a specific direction. So what happens for how does the two strands of the DNA, how are they replicated? So when they are separated, one of the DNA strands, we say it's the leading strand, while the other one is the lagging strand. So the leading strand, the DNA polymerase, because the leading strand, here's the 3' carbon, and it goes through all the nucleotides, and it ends up in the 5'. So that means that the leading strand can continuously synthesize a new strand from this old strand, from the original strand. So from this template. So this strand, the leading strand, is serving as a template for the new strand. So the DNA polymerase can very easily and continuously replicate the new strand going from the 5' to the 3' direction. However, now the other strand, you see that the nucleotides go from the 3' to the 5' direction. So that means that the polymerase, the DNA polymerase, cannot replicate this strand continuously because the nucleotides, here's the 5' of one nucleotide and it ends up in the 3'. So that means that the DNA polymerase has to replicate the new strand in different fragments. So it does one fragment and then the next fragment and the next fragment. Each fragment here are known as the Okazaki fragments. So the lagging strand is used as a template in a discontinuous manner. So the fragments are generated one at a time. And then you need a DNA ligase to ligate all these fragments to make now a continuous DNA strand. So the leading strand allows the polymerase to generate a new strand in a 5', 3' direction. Now, the lagging strand, you have one fragment made, and then it goes back and makes another fragment, and goes back and makes another fragment. So the synthesis is discontinuous, and each fragment is an Okazaki fragment, and then these fragments need to be ligated by the DNA ligase. So the lagging strand, the replication of the lagging strand is more complex. Oh, are you seeing what I'm, do you see what I'm showing? Yes?
Yes, we can see it, but I think you skipped a slide.
Okay, here we are. So you understand what the 3', 5' direction is. And now here you see how the DNA polymerase replicates one strand in the continuous manner and the other strand in the discontinuous. So the lagging strand, the replication of the lagging strand needs the DNA polymerase, but also a DNA ligase to ligate the Okazaki fragments. Okay, so the DNA polymerase is very busy because you have this huge molecule, and then you have the DNA polymerase synthesizing sections at a time. And because the process is so fast, the DNA polymerase can make mistakes. So it can bring a nucleotide that has the wrong base in sequence. So let's say here's the template, wait, 5' to 3'. This is the template, so the original strand. And here is the new strand that is being synthesized by the DNA polymerase. So you see the base pairs are complementary, CG, TA, GC. Now remember, DNA, there's no uracil. Okay, so here are the nucleotides. And they have phosphate because that's the source of energy to allow DNA polymerase to work. So the DNA polymerase is adding one nucleotide at a time. So here you see CTG. Now the next one would be thymine. Okay, and it continues on replicating the new strand from the original strand. But now here it makes a mistake. So here in the original strand, you have thymine, and now it's picking up a nucleotide that is the wrong one. Here it should be the nucleotide that has adenine, but now it picked up the nucleotide that has guanine. So it made a mistake. So, and this happens all the time. Sometimes this is what leads to mutations. Okay? So there has to be a process that will correct for this, because if not, the DNA, the new DNA molecules would be filled with mutations. So there has to be a proof reading process that can correct the mistakes of the DNA polymerase. So how does this happen? So the first thing is that there are enzymes that will cut out the segment of the DNA, the new DNA strand, that was replicated in an error, in the wrong manner. So these enzymes are known as endonucleases. Why? Because they can cut out the piece of DNA of the new strand that was wrongly replicated. So there's more than 50 of these enzymes, endonucleases. Why endonucleases? Because they can cut nucleotides within the DNA, the new DNA molecule. And then there's a DNA polymerase that is involved in DNA repair that will correct for that piece of those nucleotides that were wrongly synthesized, and then we have a DNA ligase. So the repair requires, the DNA repair requires endonucleases, then again the DNA polymerase and DNA ligase. And you will understand better here. So let's say this is the new strand that was synthesized in the 5', 3' direction. And here is a mistake. So the wrong nucleotide was introduced. So then you have an endonuclease that's going to cut out the wrong nucleotide. So then you have a space here, a gap. And this gap, because this nucleotide is going to be removed, so there's a gap here. So you need to fill in this gap. So then you have the DNA polymerase that is going to now synthesize a small DNA portion that will fill in the gap. And then you have the DNA ligase that will make this new strand continuous. So this is an amazing process that prevents mutations. Sometimes these mutations are not repaired. These mistakes from the DNA polymerase are not repaired, and this leads to mutations. So during the DNA repair, the segment that was wrongly replicated is going to be removed, excised by endonucleases. And then, so that's excision of the damaged DNA. Then you have the DNA polymerase that will fill in the gap, and the ligase that will seal the process and make the DNA, the new DNA molecule, continuous. So this process is very, very important. And there's more than 50 of these endonucleases because they are specific. They are nucleotide specific. So the DNA polymerase mistakes leads to mutations. So what can happen? A point mutation. That means it just one single base nucleotide that is substituted. And this, for example, is what leads to sickle cell anemia. So what happens, here's the original hemoglobin, and you see here this sequence of nucleotides, CTT, that will give rise to a codon for the messenger RNA, so it's complementary, GAA, and this codon is specific for hemoglobin, for glutamate. So this is the normal hemoglobin. Now let's say the DNA polymerase makes a mistake, and instead of adding a T to the original DNA, now it has an A, an adenine. So now the messenger RNA is going to be uracil. And now GUA is another codon, it's different from this codon, and it will introduce a valine. And this valine is what leads to the formation of this hemoglobin that is abnormal and it crystallizes. So here you can see the effect of just 1 point mutation, one single nucleotide change that has a major effect. So this is a mutation in the primary structure of hemoglobin. So instead of having the sequence here, this at position 6, this is the amino acid at position 6 of hemoglobin. Instead of having glutamate, which is the normal amino acid at position 6, now it has valine, and this causes havoc. The hemoglobin will crystallize because it doesn't have the proper amino acid. So this is what a point mutation leads to. Then you have deletions where some nucleotides can be deleted, or translocation between 2 chromosomes, or inversion. So these are just some types of mutations that occur. So this is all due to mistakes that the DNA polymerase made, and that the repair system is not able to correct.
Just that.
Yes. So here you have, so the question is it only a glutamate to valine mutation that causes sickle cell anemia? Yes, there could be other mutations, but this one has a major effect in the sequence, in the structure of the hemoglobin. Okay, now let's move on. So having this sequence of DNA that is so specific, we can take advantage of this specific sequence of nucleotides. So amazingly, we all have 99.1% of our DNA is similar, almost identical, not identical, but very similar among all of us. It's a very small percent of our DNA molecule that is different among individuals. So 0.9% of the DNA molecule of all those 6-foot DNA molecule, only 0.9% is different from individual to individual. So we can take advantage of that difference to use for DNA fingerprinting. What does that mean? That means that based on that difference, that small difference, one, we can identify that that DNA belongs to individual A or to the individual B, based on that 0.9 difference between the two individuals. So how can we distinguish that? So you need a very, very small amount of DNA, like a hair or a drop of blood. And this is based mostly on to perform DNA fingerprint. It's based mostly on introns. Remember that the introns are the sections of the DNA that give rise to messenger RNA, and that the introns are removed by the spliceosome. So So what happens is these introns differ from an individual to another individual. For example, an individual can have 7 repeats of these introns, while another one can have 8 repeats. So that's a big difference in terms of DNA molecule. So what happens during DNA fingerprint? So we use those enzymes that will, we extract the DNA from a small sample of blood, and we use those endonucleases to cut the DNA into pieces. Then we separate these fragments of DNA on what we call a gel, where we apply an electrical current that separates the fragments depending on their size. And so you see here individual A has this format or number of introns. Remember, the introns the number of introns in a specific section of the DNA can vary. But the individual B has other introns. So now this is the sample that we didn't know to whom it belonged, if it was to individual A or to individual B. So by running on this gel and identifying the introns that are present, we can identify to whom this sample of DNA belongs to. So you see here that this intron, these fragments match individual A. So that means that that sample of DNA, to whom we didn't know to whom it belonged, now we can comfortably and with confidence say that that blood belonged to individual A. So it's based on this 0.9% difference that separates one individual from the other. Okay, so another application of this DNA sequence and replication is that why is it that we, from a very, very small sample of blood, so the amount of DNA that is isolated from this small sample of blood is going to be minimal, and it couldn't be used alone to make any kind of reaction or any kind of identification. So this small amount of DNA that is isolated from the blood needs to be replicated. You need to make many, many copies of the DNA from this small amount of DNA. So for that, we use this kind of reaction that is known as polymerase chain reaction. And it was invented or discovered or invented by Dr. Mullis. He won the Nobel Prize in 1993. So what was he able to perform? He was able to perform from the replication. So remember replication, the copy of a DNA, a small amount of DNA. So he was able to replicate the DNA, the small amount of DNA, many, many, many times, thousands of times, artificially. So he had a cocktail of chemicals that was able to replicate the DNA strand many, many times. So how does that happen? So here's the drop of blood. You isolate the DNA. So he used this polymerase chain reaction. So it's an artificial mechanism to replicate the DNA molecule. So the DNA is extracted from a small amount from this blood sample, and then you need to separate the strands. So in this kind of reaction, you don't use helicase and all those enzymes. You just heat up the DNA, the double-stranded DNA. So by heating the double-strand DNA to 90 degrees centigrade, the strands break apart. So then primers are used. Remember, the RNA primers are essential for the DNA polymerase to work. So now you have the strands separated, and now you add the correct RNA primers that are going to allow the DNA polymerase to work. Now, to this chemical reaction, you add the DNA polymerase. But it's a DNA polymerase that's special. It can work at 70 degrees centigrade, which is not, of course, the body temperature. The body temperature is 37 degrees. So here we have a transition of temperatures going from 90 degrees to induce the separation of the two double strands, the two strands. Then you lower the temperature so that you can add the RNA primers. And then you increase the temperature to 70 degrees centigrade and use this DNA polymerase that is active at 70 degrees. So now this DNA polymerase can start working and making many copies of this small amount of DNA. So you put it in this, what we call PCR machine. That's what we use for COVID to determine if a person has COVID or not. So you use this polymerase chain reaction that allowed for a small amount of DNA extracted from an individual to be copied and copied and copied to see if the viral DNA was present. So you see here from one very small amount of DNA molecule, using the polymerase chain reaction or PCR, you can generate millions of copies using this artificial kind of sequence of chemical reactions. So the heat, the 90 degrees separates the two strands. Why do you need so many strands? You need so many strands, for example, to do DNA fingerprinting, because if you just have one molecule of DNA, you cannot use it for any enzymatic reaction, for DNA fingerprint, and so on. So you need to make many copies. So you need the two strands to make the whole DNA molecule. Okay, so you see that from one DNA, so we're not replicating, in this case, you're not replicating the entire DNA molecule, just one region, the region that has those differences. So you have the DNA primers, and now you make two. From one, you make two DNA molecules, and then four, and it's exponential. So you end up with all these copies of the DNA, of that region of the DNA. Then this can be used for DNA fingerprinting to detect if there are any viruses there, because the viral DNA is also going to be replicated. Okay, so knowing all this information about DNA, there's several applications. I told you about the fingerprinting and about DNA polymerase chain reaction. Now, another very important application is genetic engineering. What does that mean? That means that we can produce segments of the DNA molecule and introduce it into bacteria or viruses, so that then we can have the protein that is encoded by that section of human DNA to be made. So, for example, that's what genetic engineering means. That means that we are engineering a DNA molecule from a bacteria or a virus. So you introduce that human section of the DNA, and that DNA, that human DNA, is going to be replicated with the bacterial or viral DNA. So let's explain this better. So one of the techniques is gene splicing. So you know that diabetic patients need insulin. So it used to be that the insulin was very hard to produce, and it had to be isolated from different sources. But now we can make a factory that produces insulin. So a factory uses the bacterial plasmid. So this is a plasmid, the DNA from a bacterium, and you introduce a gene that makes insulin into this bacterial plasmid, and now this plasmid can make huge amounts of insulin because it has the gene, the human gene, introduced into the plasmid. So how does that work? So here's the bacterial plasmid. So you see here a bacterium that has its chromosome, and then it has these plasmids, these circle fragments of DNA. So we can use one of these fragments, this circle plasmid. We cut it with the endonuclease. Then we put in the fragment of human DNA that produces. So this is the gene that encodes for insulin. So you have to know the sequence, the nucleotide sequence of the DNA that makes insulin. So we know that very well. So the blue represents the human gene, the human DNA that produces insulin. So we're cutting the plasmid, the bacterial plasmid with endonucleases, remember? And now we put in the human gene, and we have a ligase that will ligate the human gene. So you see here, this is the bacterial plasmid, and it has added the insulin gene. So now we can put this in containers and induce the replication of this bacterial plasmid that has been transformed. So this process is known as transformation, because you're transforming the bacterial plasmid to include the human gene for insulin. So then you put it under conditions that the bacteria will grow and will produce huge amounts of insulin. So this human insulin is genetically engineered into a bacterial plasmid. Yes, will be tested with anything I say. You can use one strand of DNA for DNA print, fingerprint. Yes, but you need to make it. So once you cleave the DNA, then to use the double strand is much easier, because the one strand would be very, very small. So it's preferable to use the double strand. Yes, so all this is done in vitro, of course. You have containers like these containers, huge containers that have sort of a soup where that bacteria will be very happy to grow. So you put those bacteriums in these containers with the appropriate chemicals and cultures. So the bacteria will reproduce and will produce this genetically engineered insulin. So this seems very easy, but it isn't, because now you have to know how to purify the insulin from these baths or soup that contain all the bacteria. Yes, so we are using the bacteria as a way of producing a factory to produce insulin. Exactly. Yes, it contains the bacterial plasmid. So now that's the next challenge, which it has been figured out. How to separate the insulin from all this, the container that contains the culture, the bacterium, and all this. So that had to be worked out, that we are able to, because of the size and the properties of this protein are different from the bacterium. So you can purify it from the bacterial proteins. Okay, so another process for using genetic engineering is gene therapy. Now, instead of using bacteria, now we're going to use viruses. And what's the advantage? The advantage is that the viruses, they can integrate their DNA into the host DNA. So if you can engineer a virus DNA to contain a human gene and infect the humans, then the virus will get into the cells, into the human cells, and will integrate its DNA into the human DNA. So that means that the cells that will divide, the human cells that will divide, will have the viral DNA that contains the introduced, the genetically engineered human gene. So what is application? So let's say that an individual, for example, here, this child had a mutation in this enzyme adenosine deaminase, which was very important for the immune deficiency, for the immune system. So what happened is that a virus was made that had the gene, the human gene for ADA, that was not mutated. So then that was introduced into the patient. So the patient received lymphocytes that had the correct sequence of DNA for ADA. So that means that it was genetically engineered to express the correct form of the gene and not the mutated. And this can be used by using viruses. So viruses have the advantage, it's very clear, that they can integrate their own DNA into the human DNA. So this process is called infection because you're infecting the individual with the virus. And of course, those viruses have to be beneficial. They cannot be toxic because if not, you would kill the individual with the virus infection. So the viruses are also manipulated so that they don't cause infection. But all this looks very easy. But the gene therapy is still not used in a very effective manner. Doesn't the virus DNA end up getting killed and removed from the body? How does the DNA? So what happens is that the viral DNA gets integrated into the human DNA. So it cannot be removed. It's integrated. So you have the human DNA now that has a small portion of the viral DNA. Okay. So remember, the cystic fibrosis had a mutation that didn't allow for that chloride pump to be transported from the Golgi to the plasma membrane. So one strategy was to introduce into a patient with cystic fibrosis by using a virus to infect the cells with the proper gene for that chloride pump so that it would end up in the plasma membrane. So this was tried in a patient, but it didn't work so well. So don't infected cells end up dying. So that's the thing. The virus is modified so that it doesn't cause damage, it doesn't cause disease. So all this is a lot of genetic engineering that is involved. Genetic engineering to modify the viral particles and genetic engineering to introduce the non-mutated gene. All this is, it seems like very easy when I'm explaining, but it takes a lot of manipulation of genetic engineering. Okay. So another application is to introduce the gene into the stem cells, into the embryonic stem cells. So now those cells that are the egg and the oocyte, when they form the egg, they will have that gene that has been introduced. And now all the cells will express that gene that was genetically engineered, introduced into the embryonic stem cells. So this experiment was done with a rabbit that was albino, and the gene for a green fluorescent protein that is produced in algae was introduced into the genome of this embryonic stem cells. So all the cells expressed the green fluorescent protein. So this rabbit, when you had the blue light shining, the whole rabbit fluoresced. So because the cells expressed the green fluorescent protein. So this rabbit was albino, so it was named as Alba. So this was made to verify, to validate this process where we can introduce a gene into the embryonic stem cells, and that gene will be expressed in all the cells of that organism. Okay, so now we'll go through cell division. I'm going to go a little bit late. I'm going to be late along in this today's class. So the cell division involves the nuclear division, which goes through mitosis or meiosis, and the cytoplasmic division that is known as cytokinesis. And I know that you covered this in the lab, so I'm not going to go into great detail. So I just want to make some points that are very important. First of all, there are two types that the nuclear division can occur, mitosis or meiosis. So remember that the DNA replication occurs in the S phase of the cell cycle. So it doesn't matter if it's meiosis or mitosis. This nuclear division only occurs once. That means that the DNA replication occurs only once. It doesn't matter if the nuclear division goes through meiosis or mitosis. So the difference between meiosis and mitosis is that in meiosis, there's two cell divisions, and in mitosis, there's only one cell division. So what is the process? What is the result of having two cell divisions or one cell division? So when you have the DNA replication, you make two copies of the DNA double strand. So if you have two cell divisions, then each of the cells will receive only one copy of the DNA molecule. So that means it's haploid. It has only 23 chromosomes in humans. And this happens in the egg and the sperm. Now, if there is only one cell division, then the two cells will receive the diploid number of chromosomes. So that's the big difference. But what is important here that I want to emphasize is that in meiosis and mitosis, there's only one DNA replication. The fact that there's two cell divisions or one cell division, that's what affects the number of chromosomes resulting from meiosis or mitosis. Okay, so this is sort of a repetition of what I already said. So from DNA replication, you have one chromosome. So in humans, we have 23 pairs of chromosomes. So one chromosome before DNA replication. After DNA replication, one chromosome gives rise to two chromatids. And we call them chromatids when the two copies are connected by the centromere. So remember, I'm representing here just one chromosome, but we have 23 pairs of chromosomes. So this has to happen to each chromosome, of the 46. So one chromosome, when it's replicated,
Professor,
yes?
I'm sorry to interrupt, but I thought that together they make the chromosome and the individual ones are called chromatids.
Well, let me explain. Before replication, right, we have 46 chromosomes. So each chromosome before replication is represented as one chromosome, right? Now, this chromosome, like the other 45, are going to be replicated, right? After DNA replication. So each chromosome, there's going to be two copies of one chromosome. And while they are maintained together, those are known as chromatids. So the chromatids, each chromatid is the exact copy of chromosome, of this chromosome. So because they're linked by the centromere. Now, when you go through mitosis, then each chromatid is going to be separated and give rise to two chromosomes. Okay, is this clear? So from one chromosome first, after DNA replication, before going through mitosis, the chromosome, after DNA replication, from one chromosome, you get two chromatids. And after going through mitosis, these two chromatids, the centromere is going to disappear, and these two chromatids are going to separate and give rise to two chromosomes that are identical to this first one. You understand this? Is a chromosome paired with the centromere. So the centromere is what keeps together the result of the DNA replication of this chromosome. So each chromatid is identical to the initial chromosome. And while they're kept together by the centromere, they're known as chromatids. So then they're going to separate going through the mitosis process. And then once the chromatids are separated, now they are called chromosomes. Okay, now let's move on. So most cells go through the whole cell cycle. So G1, S phase replication, DNA replication, G2, and mitosis. So that means that most cells in our body can divide, one cell giving rise to two cells. But some cells, they move out of the cell cycle and they no longer replicate, they no longer divide. So they go into a stage that's known as the G0, where there's cell cycle arrest. So these cells no longer divide. So that's it. So if one of these cells is damaged, there's no replacement. So what happens? Which cells are these? So you see here many cells, red blood cells, connective tissue, epithelial cells, some of the muscular cells, but two types of cells, they go into G0. That means they never divide. Once they go through the cell cycle once, then they exit the cell cycle and go into G0 phase. That means they no longer divide. So if they are damaged, they no longer are replaced. And what are those cells in our body that once they go through the cell cycle once, they go to G0 and they no longer divide? So the neurons, the nerve cells in our body, and the cardiac myocytes. So if the neurons die or degenerate or damaged, they no longer are substituted. And that's what leads to neurodegenerative disorders, because certain neurons in particular areas of the spinal cord or of the brain, if they die, they cannot be replaced because they are in G0 phase. These cells, once they differentiate, the architecture is so complicated that they lose the ability to undergo mitosis. The same for the cardiomyocytes. So once they die, you cannot replace them. And that's why you have to have heart transplants, because you cannot regenerate a heart, because the cardiac muscle cells are so complex, the architecture is so complex that once they are formed, once they differentiate, they lose the ability of undergoing mitosis. Okay, so what controls cell division? Well, there are many factors. There are maturation promoting factors, growth factors. So the cells have to be healthy to divide. But what happens if there is uncontrolled cell division? So if the cells keep dividing in an uncontrolled manner, so this is what leads to tumors or growth, or neoplasm. So when the cells divide and they are in contact with each other, there's something called contact inhibition. So there are proteins that are decorating the plasma membrane of these cells. They continue dividing. Once they contact each other, those proteins that are decorating the plasma membrane, they stop dividing. That's the signal for them to stop dividing. That's known as contact inhibition. What happens if that ability of having contact inhibition is not working, if those proteins are not there? So the cells continue dividing and dividing and lead to cancer or to growth. Not all this uncontrolled cell division doesn't lead to cancer all the time. Sometimes they're benign tumors. If the cancer means that the tumors can escape and they can be metastatic, so they go to other parts of the body. So what leads to the formation of tumor growth factors and neoplasm is the lack of this signal. There's no contact inhibition. So there's benign tumors, and these are easy to remove by surgery, for example. And there are malignant tumors that are known as cancer. And they can increase in number, or they can go through metastasis. That means they spread throughout the body because they don't have contact inhibition. So,
Can you explain contact inhibition again?
so contact inhibition means that in each cell, right, this is a cell, and that's the plasma membrane. So on the plasma membrane, you have these proteins that are involved in this signal that's known as contact inhibition. So if one cell contacts the other and these proteins are contact each other, they send signals into those cells and they say, stop dividing. You understand what I'm saying? So let's say this cell, right? This is a epithelial cell, makes up, for example, the skin. So outside on the plasma membrane, you have proteins that you don't see here that are involved in this contact inhibition signaling. So if there's another cell here that touches this cell, then that signal, that contact inhibition is activated and the cells will stop dividing. If that contact inhibition is not there, then that leads to tumors. Some are benign and can be removed because they stay in a certain place. Others are malignant.
But why does like if it touches another cell make it divide more? Like why is that what happens?
No, if you don't have contact inhibition, if that signaling is not working, then the cells will continue to divide. See, in this case, the contact inhibition is not working. The signaling that is induced by contact of the cells, one contacting each other, it doesn't matter, it's not working. So the cells will keep on dividing. So how do you prevent this? Well, that's the very important question. You can treat cancer, you can treat with chemotherapeutic agents in all kinds of ways, but that's what happens. That's what the question is. How can you stop the cells from growing in an uncontrolled manner? I mean, from dividing. So let's see what are the stages of metastasis. So that's the worst case scenario, when the cells continue to divide and they spread through the blood cells and they invade other areas of the body. That's what metastasis is. So there's 3 steps. First is the invasion. So let's say you have a growth. So these cells are abnormally dividing and they form a growth. And some of them escape and go into the blood vessels. So you go through invasion, means that they separate from the primary growth. Then there's penetration. That means they go through the blood vessels and then escape. They escape from the blood vessels and go to other areas of the body. For example, here you have a case of metastatic prostate. So you had the growth in the prostate, and those cells escaped the prostate, they went into the blood vessels, and they traveled to the bone. And now they stay in the bone, and they continue to divide in the bone. So that's what happens. These cells were started, these cells that lost contact inhibition, and they start dividing in an uncontrolled manner. They escaped the original tumor in the prostate, and they went into the blood vessels and into the lymph nodes, and they invade other tissues in the body, such as in the bone. So here you see the prostate, and the cells went all the way now to the lungs. So that's what metastasis is. So you have cells that escape from the original growth, go through the blood vessels and the lymph nodes and the lymphatic vessels, and they lodge themselves in other parts of the body. And that's what the bad news is. So talking about types of cancers, there are carcinomas. That means they are cells, epithelial cells of the skin. Melanomas, they're also cancer of the skin, and these are the most dangerous ones. They affect melanocytes. So when melanocytes, which are the cells in the skin that produce melanin, that give color to our skin, when they start dividing in an uncontrolled way, they can go through metastasis very quickly. So these are melanomas, or cancer of melanocytes. Then sarcoma, they are cancer of muscle and connective tissue. Leukemia is cancer of the white blood cells, and lymphoma, cancer of the lymphatic tissue. So what is here, what do these have in common, is that the cells lose the ability of controlling mitosis. So they keep undergoing mitosis for unknown reasons. It could be environmental. It could be that they have mutations. It could be virus. Many times most of the time we have no idea why these cells lose the ability of controlling mitosis. Yes, that's what differentiates the different stages of cancer. So it's how the growth escape, how it spreads to different parts of the body. So what are the causes of cancer? Carcinogens, chemicals that are present in the environment. Smoking also can be a carcinogen. Viruses also can cause cancer. And now we have some genes that mutate and now induce cancer. So they prevent the cells from controlling mitosis. So these genes are known as oncogenes. For example, HER2 and c-MYC, these are oncogenes. Why? What are oncogenes? Oncogenes are genes that transform cells, that prevent cells from controlling mitosis. So that's what an oncogene is. An oncogene causes cancer. It prevents the cells where these genes are located to have controlled mitosis. Then we have proto-oncogenes. What are these? These are genes that are usually normal, but all of a sudden, for unknown reason, they become mutated, and now they become oncogenes. For example, breast cancer, you have BRCA1. So BRCA1 is a gene that causes cancer, breast cancer. But the gene, if it's not mutated, it's a normal gene. It's necessary for the cells to survive. But if it becomes mutated, then it works on the cell. It prevents it from having regulated mitosis. So a proto-oncogene is different from an oncogene. The oncogene, this gene per se, has the intrinsic ability to cause cancer. A proto-oncogene is a normal gene that does not cause cancer until it's mutated. That's the case of BRCA1. And then there's some genes that prevent the the oncogenic effect of these genes. So one gene that is very, very important, that is protective gene, is P53. It's a tumor suppressor. So it prevents the effect of these oncogenes. And actually, P53 is subjected to mutation. 50% of cancer cells contain mutant P53. So that means that the P53 gene is no longer able to suppress the tumor. Okay, so what are some examples of cancer treatment? So removal by surgery, that means if the cancer has not yet spread throughout the body, so it's in a specific area, it can be removed by surgery, by freezing the cells. If you freeze the cells die, or by radiation. So if this is not possible, then you have to kill the metastatic cells. So that means that the cancer cells are no longer present just in one area. So they cannot be removed by surgery because they're all over the body. So now we have to use other strategies. So chemotherapy, this is using chemicals that are toxic to the cancer cells. And chemotherapy is very hard to establish because it depends on the concentration. So you're giving the patient chemicals that are toxic. So they're toxic to all cells. But what happens is, because I already explained this, because the cancer cells are very active, undergoing mitosis in an uncontrolled manner, they're very, very active. So you can lower the concentration of these toxic chemicals so that they will hopefully only affect the cancer cells and not affect the normal cells that are less active. Or you can use radiation also. Yes, they are specific. For example, papillomavirus. There are certain viruses that are linked to cancer. Yes. Okay, so now we go to the integumentary system. So I know that you went through this in the lab, that you had a lot of exposure in the lab to the integumentary system. So I'm just going to, let's see, in the last 4 minutes, I'm going to end the class at 5:30. I'm just going to go through some of the aspects of the integumentary system that I think are more interesting. So for example, the skin, you have this malignant melanoma that are very, very aggressive. So so melanoma affects the melanocytes. The melanocytes produce melanin. So how can you tell if you have a melanoma on the skin? So you have this ABCD kind of classification. So sometimes you have black spots on the skin. That doesn't mean that all the dark spots on the skin are melanomas. Some can just be uncontrolled production of melanin. But what you want to identify is if that dark spot is a melanoma. So here are the criteria for classifying a dark spot as being melanoma. First of all, it's asymmetric. So this, you can see that the outer surface of this area is asymmetric. The borders, so asymmetric means this side is different from this. The borders are irregular. The color is not even, it's uneven. It has black, brown, pink. And this is the major criterion, is that it changes shape and size with time. So if you have a dark spot and with time it increases, that could be a melanoma. It doesn't mean that it is a melanoma, but it's time to go and see doctor. So the skin cancer, 88, it has increased tremendously because of sun tanning. So the UV light of the sun is a cancer agent. So over the last 50 years, the incidence of cancer, skin cancer, has increased tremendously because we like to be in the sun for longer and longer time. So the sun is essential for our health, but not for long periods of time. So you know that there's the epithelium, the epidermis, and the dermis are the two layers of the skin. One interesting aspect is the epidermis has no blood vessels. So how does it get its nutrients? By diffusion from the dermis to the epidermis. So there are no blood vessels in the epidermis. If you have a scratch and just damage the epidermis, you won't bleed. So what are the functions of the skin? So there are many interesting functions of the skin, but one that I want to emphasize is that the skin in the dermis, right? Because the epidermis has no blood vessels, but in the dermis, you have 80 to 10% of the total blood in our body. So it's a blood reservoir, has many blood vessels, and there's 8 to 10% of the total blood in our body is stored in the dermis. And then I want to focus also on this function of the skin. So the skin is the site where the initial steps of the synthesis of vitamin D. So remember, vitamin D is a steroid, right? It's a lipid. It has this structure. So to make vitamin D, you need the sun. So you have these two effects of the sun. You only need about 20 minutes of sunlight to be able to synthesize enough vitamin D. If you have long exposures to the sun and to the UV light, then that can lead to cancer, not all the time. So, and you know that the melanin, why do we have melanin? It's to protect the DNA from the UV light. So vitamin D, the initial component of vitamin D is 7-dehydrocholesterol. So vitamin D is a product of cholesterol. So if you have 1 hour per week of sun, this is enough to convert the hydrocholesterol into vitamin D3. This vitamin D3 is still not active. So it has to go through the blood, takes it to the kidney. And in the kidney, it's converted to calcitriol. And calcitriol is the active form of vitamin D. And you might ask, why do we need vitamin D? We need vitamin D because vitamin D is very important for bone density. What does it do? It allows calcium to be absorbed from the digestive tract. So it improves the absorption of calcium from across the digestive tract. So that's why you need vitamin D, because this calcium that comes from the digestive tract into the blood, then it's deposited on the bone to have the bones be strong. So bone density is highly dependent on vitamin D, not the only factor, but it's highly dependent on vitamin D. Okay, and these are the different types of cells that you already know, that you've learned from the lab. And what are the pigments that contribute to the color of our skin? One thing that I want to point out that is very interesting is that we all have the same number of melanocytes. So melanocytes are the cells in the skin that produce melanin, this pigment. So we all have the same number of cells. We might have less or more melanin. And what is the advantage of having melanin? So melanin protects the DNA of the cells in our body. So melanin is made from this amino acid known as tyrosine. So there's this enzyme, tyrosinase, that is activated by UV light, and it converts tyrosine to melanin. So the darker the skin, the more protection you have to the sunlight. And if you don't have an active tyrosinase, then you don't have the pigment in your skin. You are an albino. So if the tyrosinase is not functioning, if it's mutated, then tyrosine cannot be converted to melanin. And this leads to albinism. And individuals that suffer from albinism have to be protected from UV light because they don't have the advantage of having melanin to protect the DNA in their cells. And an interesting disorder that's associated with melanin is known as vitiligo. So Michael Jackson was supposed to suffer from vitiligo. And what is this disorder? This disorder is an autoimmune disease. Remember, we talked about multiple sclerosis. It's also an autoimmune disease where the white blood cells, in this case, attack the melanocytes. So if they attack the melanocytes and the melanocytes are no longer functional, they cannot produce melanin. So vitiligo is an autoimmune disease in which our white blood cells, our immune system, attack the melanocytes. Okay, hemoglobin, we talked about this, and these are other conditions like jaundice, where you have a yellowish colored skin. It's due to activation of or high concentrations of bilirubin. This pigment is produced by the liver. If you have some problems with the liver, you have too much bilirubin that gives this yellowish appearance. Then you have cyanosis. Cyanosis gives the skin sort of a bluish coloration, and this means that the hemoglobin is not working properly. It's not bringing enough oxygen, and therefore the blood will have this blue coloration. Okay, you've gone through burns, I know, in lab, and the different phases of wound healing, where you have inflammation, migration. Okay, so I think I'm going to stop here.
Professor, I have a question.
Yes, please.
So I was looking at the lab material, the lecture materials for the exam, and I noticed that it said that the models were going to be a part of the models were going to be on the test. So I thought if I just go through the lecture slides, like, will I be good for the exam, the lecture exam?
Yes, there's not going to be any anatomy in the exam, only physiology. You only have to concentrate on the notes, the slides, and the recordings. And if you go through to I just posted.
As it said, like figure 1.6 is on it, but I was confused because
No, figure 1.6, that means you need to know the function of the different systems. And
but that's in the notes, no?
Yes, everything that's in the notes you need to know, and on that figure. Okay, so the rest of the slides is really in different diseases associated with the skin. Maybe I'll talk about. Yes.
So are we going to have in the exam all the chapters you have covered, or is going to be some specific chapters?
No, the chapters are 1, 2, 3, and 5.
Okay.
Let me just finish this with the skin cancer.
All
So
right.
remember, the cancer of the skin is loss of contact inhibition. So there's basal cell carcinoma, squamous cell carcinoma, and malignant melanoma. This is the worst because it's metastatic. So the basal cell carcinoma does not metastasize, and it's associated with the epidermis only, not the dermis. Then squamous cell carcinoma, it's still associated with the epidermis, and it does not metastasize. And now you go to malignant melanoma, and that is associated with the melanocytes. And this can kill because it's a very, very aggressive form of cancer. Okay, so I finish here. So the chapters are 1, 2, 3, and 8. And in our review on Tuesday, I can go over some of this material that I did not have time to cover today. But it's all in the lecture notes and on the slides.
Professor, I don't think we have class on Tuesday though.
That's right. That's why I have a review on Tuesday.
Okay.
So the first slide on today's lecture, it has the announcement that I'm going to have a review session instead of class. No one has class, but I'm going to have a review session from 4 to 5:15 on Tuesday. I know that and it's not going to be recorded, some students cannot attend it because of religious causes. So please email me and maybe we can work something out.
Professor, it will be on Tuesday, right? The review?
Yes. Tuesday from 4 to 5:15. Just pretend that we have in class. You don't have class, but we, instead of class, we having a review because the exam is on Thursday, right? So this first exam, this first slide is very important. You should look at it in the announcement. So we have a review session on Tuesday, September 23. There's no class, there's no labs, there's no lectures, there's no class in the whole Hunter, but we having a review session. Also, I posted on our Bright Space website examples of questions for exam 1, and also what's going to be on the exam, the material for exam 1. So you should go there and click on these PDFs and read them. Okay?
Professor, I have another question.
Yes.
So there was a document that you posted with like, I think it was about 83 questions, if I'm not mistaken. Are those gonna be like the main focus for exam number 1?
So those are not multiple choice, right? They're just review questions.
No, they're just like the ones that are broken down into sections, like the
Yes.
introduction, chemistry, and all.
Right. They're not multiple choice, correct?
No, they're not multiple choice,
Right.
no.
So yes, those questions are there so that once you study, you can see if you can answer those questions. They don't cover all the material, but they cover most of the material.
Okay,
Okay?
thank you.
It doesn't just work.
Professor, I gave you a question in the chat.
Yeah, I'm reading it right now. I'm assuming for the exam, we don't need to memorize any skin models or anything. Also for the exam, do you want us to memorize the nucleotides, like being able to identify the nucleotides? No. No anatomy and no nucleotides, except for the systems, the 11 body systems. You have to know their function and a little bit what are the organs that are involved there. Don't understand this. Student disabilities, please don't. I don't know what that means. Where do we find the document Brightspace? Oh, it's under review. I'm going to put it here so that it's easy for you guys to access it. I'm going to put it here in the announcement. The integumentary system is on the exam, yes. Okay. Anything else?
Yes.
Yes, please.
Okay.
Go ahead.
Yes,
What?
Professor.
Yes,
I will ask you the question in the chat. I know I have different special accommodations, so you're gonna send it over because I know you said you're gonna have you give us a password for exam
yes,
sort.
yes, don't worry.
Okay.
The proctors in that office will have the password.
All right. Okay.
No problem. Everything will be smooth. Don't worry.
All right. Thank you, Professor, and God willing,
Sure.
Tuesday.
Sure.
Bye-bye.
Bye-bye. Any other question?
Yes, one final one. So if you said to cover the slides, the notes and the recordings, and also the document that was open-ended. So if we do all of that, we should be good for the exam, right?
Yes, I don't understand what you mean the document that's open-ended.
Oh, the question I asked about the one that you posted on
Oh,
Brightspace
yes, the review, right?
and the review packet.
Yeah, I'm going to put it here on this slide, this first slide. It's in the
Okay.
notes so that you can just click on.
Okay, Professor. Thank you.
You're welcome. So I hope to see you, most of you, on Tuesday.
Professor, hello?
Yes, yes, I'm
Yes,
here.
I sent you an email about the invitation. I don't know, like I got your second email saying that you just sent me the invitation, but
Yes,
not the actual invitation to create a username and password. This is what I need. Because when
I send you the invitation. So,
yeah, I got
Wait.
the second email that
right.
said, and your second email said that you just sent the invitation and then you sent me the instructions attached to the.
I send you the invitation. You should have received it.
Well, I just don't know where I don't know because
Well,
I have a Hunter email. Well, I emailed you back anyway to show you what I received.
I know. So you should go to the Hunter email.
Well, I don't have a Hunter email like I
Yeah.
tried to explain to you because
You have to have, then you have to go.
I don't have a Hunter email because I'm taking classes via e-permit. I don't attend. I'm not.
But if you register at Hunter, you should have a Hunter email. So what you need to do is go to the help desk.
Okay, I'll call them tomorrow.
Yeah, you know what that is?
Yeah, yes.
Okay. So
Okay.
they are very, very helpful. And
Okay.
they should help you. And if you have a problem, email me again after you
Okay.
with the help desk, okay?
Okay, I will. Thank
Good.
you.
You're welcome. Yes, you need to know the diseases, yes. As I said, you have to know everything that's on the slides, on the notes, and on my recordings. Okay? Is this good enough? Can we stop now? Okay, everybody. Bye-bye. See you Tuesday. Have a good weekend.